Hello, everyone, and welcome to today's Keynote webinar titled Microbiome-Mediated Mechanisms of Immune Therapy Failure for Ovarian Cancer. This webinar is a part of the fourth annual event in the Immuno-Oncology virtual event series. I am Melanie Chan of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located within the abstract on the left of your screen and follow the process to obtain your credits. Now, let's get started. I now present today's speaker, Melanie Rutkowski, Associate Professor at the University of Virginia. For a complete biography on our speaker, please find it on the left of your screen. Melanie, you may now begin your presentation.
All right. Thank you so much, Melanie. And thank you to the organizers for allowing me to talk about our work today. And thank you all for your attention. And so today I'm going to be telling you about a story that we're working on for ovarian cancer and how we believe that we've identified a pathway where the host microbiome is contributing to failure for immune therapy of ovarian cancer. And so ovarian cancer is the most fatal gynecological malignancy. And one of the largest barriers to treating and reducing the mortality associated with this disease is that the majority of cases are diagnosed at advanced stages of disease, when the disease has progressed either within the abdomen or beyond the abdomen. And so this is why oftentimes ovarian cancer is referred to as a silent killer, because oftentimes when it's stage 1 or 2, women don't even realize that they have ovarian cancer. And so as you can see here by this 5-year relative survival, as the disease progresses and it becomes more distant, the 5-year relative survival drops precipitously. The other thing I would like to point out with this survival curve is that, you can see over the past few decades, and this is even going before 2004, that overall outcomes have more or less remained unchanged over the past several decades, despite intensive efforts to improve outcomes for this devastating disease. And so one of the potential secrets that may be, kind of underlying enhancing ovarian cancer survival are T cells. And this is illustrated by this study by Zhang and colleagues, where they demonstrated that ovarian tumors that are infiltrated with high amounts of T cells, or intratumoral T cells, the red line, have a significantly improved survival outcome compared to those tumors that have few to no T cells in them. The problem, though, is that efforts to kind of leverage the fact that these T cells are present within tumors, either using immune therapy or other immune adjuvants to kind of reboost them and re-elicit their pressure against the tumor, have really achieved very limited success for patients. And so, for example, immune therapies, for example, PD-1, PD-L1 blockade, which are used to essentially relinquish the immune suppressive pathway of T cells, enabling them to re-exert function against tumors, have really achieved low overall response rates. This is exemplified by studies that have actually tried to combine then immune checkpoint blockade with other elements such as chemotherapy or other inhibitors that are targeting other tumor microenvironmental factors to reduce immune suppression. But yet again, it did not demonstrate a significant improvement in patient outcomes. Results from larger clinical cohort studies, such as the Javelin and the Imagine trials, have actually indicated that the metrics that we typically use to assign parameters to predict responsiveness to immune therapies for other solid tumors cannot be extrapolated to ovarian cancer. And so this suggests that there are potentially undefined immune suppressive pathways that are unique to the ovarian tumor microenvironment that may be contributing to failure of immune checkpoint blockade. Thus, there's a really critical need to define novel therapeutic strategies that can enhance anti-tumor T cell function and therefore the outcomes for patients with ovarian cancer. And so why are T cells so dysfunctional? Why is it so hard to get them to work within the ovarian tumor microenvironment? And so this is another barrier, is there are multiple cellular barriers within the ovarian tumor microenvironment. And so here on the left, you can see many non-immune barriers that are present within the tumor microenvironment, including adipocytes, fibroblasts, mesenchymal and epithelial and endothelial cells, not to mention the tumor itself. These cells are producing multiple metabolites and other immune suppressive factors that not only enable the ovarian tumors to grow, but they actually exert a profound and negative impact on the anti-tumor immune response. And so many of these metabolites will directly impair anti-tumor T cell function, but it will also lead to the accumulation of other suppressive subsets, such as suppressive dendritic cells, macrophages, and immature tumor-associated neutrophils and myeloid-derived suppressor cells, all of which work together to create this really profoundly hostile environment for T cells within the ovarian tumor microenvironment. And so in terms of the studies that have worked to kind of understand and overcome some of these cellular barriers for ovarian cancer, most of the effort has gone towards identifying tumor-associated factors that are leading to a more metabolically and immunologically hostile tumor microenvironment. And so our laboratory is interested in identifying host intrinsic pathways that may also contribute to T cell dysfunction in ovarian cancer. And we believe that this is related to the host microbiome. And so how is this related to the microbiome? We believe that we've identified a pathway involving toll-like receptor 5 signaling. So this is based upon work that I had done as a postdoc, where we identified that TLR5 mediated recognition of gut microbes that are finding their way into the ovarian tumor microenvironment are actually leading to the increase in very tumor promoting factors. This enables the accumulation of suppressive myeloid cells into the tumor microenvironment and inhibition of anti-tumor T cell function. And so in the survival graph on the right, you can see that wild type animals have a significantly reduced survival outcome compared to mice lacking toll-like receptor 5 using an orthotopic ovarian tumor model. And so taking a step back, what are toll-like receptors? And so you can kind of think about toll-like receptors as a zip code, allowing the cells within our body to recognize and distinguish between unhealthy dying cells or pathogens compared to healthy cells so that they're not attacking our cells. These receptors are typically expressed on cells that are either surveying the body to protect against pathogens and to recognize and to clear dying cells. They're also found on cells at these barrier surfaces, which essentially comprise the first line of defense when pathogens are trying to enter into our body. Now, they're found both on the surface and inside of cells, and their location really depends and is specific to the types of ligands or molecules that they're actually able to recognize, oftentimes organized according to where they may encounter these pathogens. So for example, the ones on the surface of the cell tend to recognize and be more specific towards bacteria, certain fungi, and parasites that don't tend to invade inside the cells. Whereas the ones on the inside of the cell tend to recognize for pathogens that can invade, such as bacteria, and viruses. And so what happens then when these receptors are engaged? Canonically, it's thought that these receptors, when engaged, will transmit a signal into these cells that are expressing the receptor. and it will activate them. It will turn them on and engage them so that they can eliminate the pathogen or the cell that's infected by the pathogen. Then they will go on and induce a robust T cell response. And so traditionally, they are associated with the induction of anti-tumor T cell immunity. And as I mentioned, our lab is interested in toll-like receptor 5. And so toll-like receptor 5, the only known ligand for TLR5 is bacterial flagellin. And so that's kind of these tails on bacteria that kind of help them move around. However, there has been recent studies, including studies from our lab, that have indicated, especially in the context of cancer, that unlike other TLR pathways, TLR5 signaling may associate with a more immune suppressive environment. And this kind of is aligned with recent studies that have demonstrated that chronic TLR signaling can actually lead to cancer progression. And so we believe that understanding how TLR5 signaling influences tumor progression in response to immune therapies really has the potential to unveil therapeutic or mechanistic pathways that we can use to target that involve either the commensal microbiome that help us to create therapies for these solid tumors that are occurring distally from barrier surfaces. TLR5 signaling actually has significant clinical relevance as well. And so approximately 8 to 10% of the general population harbors a germline mutation in toll-like receptor 5. And so what happens is it's a single nucleotide polymorphism that introduces a premature stop codon right within the flagellin binding domain. And because TLR5 signals as a heterodimer, even having just one copy or a heterozygous carrier leads to functional consequences in that these individuals are unable to recognize toll-like receptor 5 ligands. And so you can see here these PBMCs, or peripheral blood cells that are stimulated with bacterial flagellin, are unable to respond, and these are heterozygous carriers. However, their response to LPS remains intact. For ovarian cancer, this also has clinical significance. And so we find that within the 10% of ovarian cancer patients that have this polymorphism, if they are diagnosed with advanced stage ovarian cancer, there is a significant survival benefit to having this polymorphism or to being deficient in toll-like receptor 5 signaling. And what we have shown in the past is that having this polymorphism not only leads to improved survival outcomes, but we have evidence to suggest that the immune environment is also distinct and potentially less immune suppressive, thus more amenable to immune therapies. And so to dig into this a little bit more deeply, we turn to the GPA database, where we can extrapolate levels of toll-like receptor 5 expression within ovarian tumors. And we quartiled them and compared the high versus the low TLR5 expressing ovarian tumors. And you can see in this graph in the left that having low TLR5 expression within ovarian tumors corresponds to a significant survival outcome, especially with the long-term survivors. However, there is no survival benefit for toll-like receptor 5, indicating that not all TLRs are equal, and that TLR5 expression may have a unique and negative effect on ovarian tumor progression, unlike other TLRs. And so how would bacteria actually even get into the tumor microenvironment, since ovarian tumors tend to not happen in the gut? And this evidence and clues about this come from a recent study in cancer discovery by the Zippelgil lab, where they showed that multiple extraintestinal tumors produce molecules that actually cause damage at the gut surfaces, leading to a state of imbalance and dysbiosis and gut leakage. And so this can actually easily be measured by feeding mice with these inert fluorescently labeled dextran beads. And because the beads cannot absorb through the gut, when it's intact, they'll be passed into the feces. However, when there's breaks within those junctions or cell death and breaches at those surfaces, the beads will find their way into the blood, where we can measure them by the fluorescence. And as you can see in the dot plot below, that ovarian tumor-bearing mice have significant levels of these beads present, indicating that the tumors are indeed causing gut leakage. We went a step further and were able to actually measure the presence of these ligands within the tumor microenvironment, either using a reporter assay, which is the top bar graph, or using universal primers to detect for the presence of bacteria. And in both scenarios, you can hopefully appreciate a significant amount of TLR ligands within the ovarian tumors and the tumor microenvironment compared to non-tumor bearing animals. And so this indicates that there's this signaling pathway that is out of context, that is not present in a healthy setting. And we believe that this is leading to the overamplification of these tumor-associated factors and T-cell inhibition. And so we wanted to ask, what effect does the TLR5 signaling actually have on response to immune checkpoint blockade for ovarian cancer? As I mentioned before, trials have really indicated a very poor and dismal response to checkpoint blockade. And so we use two orthotopic models where we transplant them into the tumor microenvironment, very much recapitulating stage 3 for advanced stage disease. And you can appreciate that looking at the solid red line, that when you give TLR5 deficient animals immune checkpoint blockade, there is a significant survival benefit. Importantly, and corresponding with the human data from the GPA cohort, in TLR4 deficient animals, they do not receive a benefit from immune checkpoint blockade, again, demonstrating that this is unique to the ovarian tumor microenvironment. We next wanted to test the durability of the survival response to see whether there's a memory response that is initiated in these TLR5 deficient animals. And so we re-challenged the survivors after 100 days with the same tumor cell line in the absence of therapy and monitored their survival. And looking at the solid red lines, I hope you can appreciate that, again, we saw a significant survival benefit. And the fact that animals are dying even after 300 days really indicates long-term ongoing pressure against the tumors that is eventually lost, likely due because the animals have old age. We also used a more clinically relevant ovarian tumor model that is developed from the fallopian tube epithelium, which is thought to be the cell of origin for ovarian cancer. And so these tumor cells are another orthotopic model that harbor common mutations that are found in high-grade serous ovarian cancer. And again, what we find is that although the survival benefit is not as dramatic, there is a slight improved response in TLR5 deficient animals during checkpoint blockade. And so we wanted to understand what might be driving some of these differences. And so we looked at the tumor microenvironment of tumor-bearing mice during early tumor progression, prior to and during when we would be delivering the immune checkpoint blockade. And one of the cell types that came out of this analysis was that TLR5 deficient animals had an expanded population of cross-presenting dendritic cells, or traditionally called CDC1. Now, these cells are really critical for orchestrating anti-tumor immunity in various tumor settings. They can present antigens to CD8 T cells, and they oftentimes correspond with improved responses to immune checkpoint blockade. And so corresponding with this increase in these cross-presenting dendritic cells that we're finding in our TLR5 deficient animals, we also see a reciprocal increased T-cell response. And so we're looking at T-cell effector function activities. We find that our T cells at early time points are exhibiting evidence of recent degranulation, and they're producing a lot of interferon gamma. What's striking about these data is that not only is it sustained during early stages of tumor progression, but even during advanced stages at day 30 and during immune therapy, that response is elevated over what we're finding in our wild-type animals. And so our data thus far indicate that TLR5 deficient animals during immune checkpoint blockade have T cells with increased effector function. And this is leading to an improved survival outcome. On the other hand, wild-type animals, they do not see that benefit during immune checkpoint blockade. Thus, they're not receiving a benefit similar to what we're finding in our TLR5 deficient animals. And so we wanted to understand the mechanism of action of this. And to identify which cells are actually expressing TLR5 to know why our T cells are having less function in the presence of TLR5 signaling. And so to do that, what we did was we used TLR5 reporter mice. And so these animals have a TD tomato reporter that is expressed alongside of TLR5. And so any cell that is expressing TLR5 is also expressing this red TD tomato reporter, as you can see in this picture of the intestines, all of these red cells. And we use flow cytometry to look at cells within the tumor microenvironment. And what we found is that T cells are not expressing TLR5. And this is to be expected based upon what is known in the literature. However, the majority of cells within the tumor microenvironment that do express TLR5 are these myeloid cells. And so we wanted to ask, does TLR5 signaling on these myeloid cells, is it a direct effect? Or is there something within the tumor microenvironment that may be polarizing them or reducing the accumulation of these cross-presenting dendritic cells during immune checkpoint blockade? And so we used two different strategies. The first strategy we used was an in vitro culture system where we tried to expand these cross-presenting dendritic cells with FLT3 ligand plus or minus chronic or acute flagellin exposure. And so this is happening in the absence of any tumor-derived factors. We also used a second approach where, because everything we've done to this point is in separate tumor microenvironments, we wanted to put the wild type and TLR5 knockout cells into the same microenvironment to test whether they are behaving the same, because this would indicate either a direct effect or maybe some microenvironmental factor that we've missed. And so to talk a little bit about the in vitro culture, FLT3 ligand has been recently demonstrated to expand these cross-presenting dendritic cells. And so it's a really nice way to try to achieve higher numbers of these cells using bone marrow from mice. As you can see, the CDC1 cell type at the bottom left-hand corner of your screen, they express high levels of the receptor. And so we set up these cultures. And so paying attention to the red lines, you can see that the red dots, that the graph on the left in the wild type animals, the CDC ones were not really affected. However, within the cultures, what we started to observe was an expansion of cells that you don't necessarily want within the ovarian tumor microenvironment. Myeloid-associated dendritic cells, monocytes, which express Ly6C, and other myeloid cells, which are predominantly macrophages. We're not seeing this in the TLR5 knockout, which is expected, and we're not seeing this in the acute flagellin exposure of the wild-type animals. Importantly, what we found was that if we look at these expanding cell subsets functionally, we start to observe that they're starting to exhibit high levels of PD-L1 expression, indicating that they're becoming, not only we're expanding the cells that you don't want there, but they are becoming suppressive. They're expressing high levels of PD-L1, which is potently suppressive for T cells. Now, looking at the in vivo approach, the way that we set this up was we lethally irradiated congenic mice that have this marker on all of their immune cells that you can easily distinguish. We did that to enable us to engraft a 50-50 mixture of wild type or TLR5 knockout bone marrow. And because they have these different markers, we can tell which one is wild type versus knockout. We allowed these animals to recover for 10 weeks to allow full reconstitution of the immune system. And then we checked to determine whether there was equal engraftment of these cells. And as you can see by this graph that just popped up, it was about 50-50 engraftment, meaning that one population did not have or outcompete the other, which was good. And so we initiated tumors and started immune checkpoint blockade. And what we found was really striking in that in our knockout mice, despite being within the same tumor microenvironment, we still observed this expansion of these cross-presenting dendritic cells. Whereas in the wild-type animals, we started to see an accumulation of more myeloid-derived cells. Interestingly, in the wild-type animals, the cross-presenting dendritic cells that were expanding were expressing much higher levels of PD-L1 blockade. And so there were more of them, and they were also expressing it at a higher level, which is not shown within the tumor microenvironment. And we did not observe that in our wild-type animals or in our non-tumor bearing controls. We also saw a trend towards increased IL-12 production for our TLR5 knockout mice, TLR5 knockout immune cells, suggesting that despite being in the same environment, TLR5 signaling is having a direct effect on the immune system, leading to the dysregulation or repolarization of these myeloid cells, and that might be the cause of this failure for immune checkpoint blockade. We wanted to dig into this a little bit better and more deeply, and so we used an approach called site-seek to define how chronic TLR5 signaling is actually affecting these myeloid cells. And so we use the in vitro culture approach, again, in the absence of tumor-associated factors, so that we could directly test the effects of chronic flagellin stimulation when these cross-presenting dendritic cells are being expanded. And so site-seek allows simultaneous analysis of protein and RNA expression at a single cell level, really helping to provide quantitative and qualitative attributes of all cell populations, within a given setting. And what we found was within the entire bulk cultures that were exposed during that expansion to chronic flagellin, there was this upregulation in pathways that tended to associate with a very metabolically stressed cell state, that associated with impaired function, and pathways that tended to be associated with upregulation of PD-L1. There was also increased proliferation, suggesting that these cells were being retained in a more immature subtype, especially when you're comparing to the pathways that were downregulated in the non-flagellin exposed, that were upregulated in the non-flagellin exposed cultures, really indicating that, during this culture period, as expected, the cells are maturing and they're more functionally competent, through their metabolic and functional pathways. We next wanted to identify the cross-presenting dendritic cells. And so because we did not see differences in their expansion, we wanted to ask whether they are transcriptionally different, or they're just more of the bad players that are present within that microenvironment. And so we identified 18 clusters with our single-cell RNA sequencing, and we used both protein and RNA expression to identify our CDC1s, which is this cluster 10 circled here in the red. What we found with this analysis is that indeed the cross-presenting dendritic cells are significantly distinct. We find the upregulation of genes and that are associated with more immature, metabolically pathogenic myeloid cells. On the other hand, we see downregulation of genes associated with CDC functions, such as XCR1 and IRF1. And if you look at the pathway analysis, multiple pathways are downregulated with this chronic flagellin exposure, again, that associate with a more mature and competent cell type. And so altogether, these data suggest that chronic TLR5 exposure is not only expanding a stressed, metabolically and functionally impaired myeloid populations, but within these cross-presenting dendritic cells, they're also exhibiting evidence of being functionally deficient. And so we wanted to take this full circle. And so we wanted to ask whether if we create mice that lack TLR5 specifically on dendritic cells, will we see an improvement in overall outcomes during immune checkpoint blockade? And what we found, as highlighted by this solid red line, is that yes, if we knock out TLR5 specifically on dendritic cells, or all cells expressing CA11C, we see an improvement in response to immune checkpoint blockade, although it's nowhere near as robust as a full knockout mouse, suggesting that there are other cell types that are also receiving this signal for TLR5. Importantly, looking in the tumor microenvironment of these animals, we find that in mice that lack TLR5 only on their dendritic cells, we start to see increased numbers of these cross-presenting dendritic cells, and this corresponds with improved T cell function, as indicated by evidence of T cells that have recently degranulated by CD107 staining or interferon gamma production. On the other hand, in mice with intact TLR5 signaling on their dendritic cells, their dendritic cells overall tended to express much higher levels, even on a per-cell basis of PD-L1. And this tended to correspond with T cells that were exhibiting signs of exhaustion through these expression of these exhaustion markers, PD-1 and LAG-3. And so these data altogether suggest that chronic TLR5 signaling, presumably on these myeloid precursors, is leading to the expansion of these myeloid cells that are either immature or suppressive, leading to upregulation of PD-L1 at the expense of cross-presenting dendritic cells. This in turn impairs T cell function, leading to reduced response during immune checkpoint blockade. Because TLR5 deficient mice are not having this impairment, their cross-presenting dendritic cells are arriving into the tumor microenvironment. And with the boost of the PD-L1 blockade, the T cells are now much better poised to elicit effector activity against tumor cells, leading to a significantly improved survival outcome for ovarian cancer. And so, ultimately, these data demonstrate that TLR5 signaling contributes to immune suppression and failure of immune checkpoint blockade. And we've also confirmed that this is the case with CTLA4 blockade as well. And so it's not just the PD-L1, PD-1 signaling pathway. And that this chronic TLR5 signaling, when these myeloid cells are expanding, really impairs the differentiation and functional attributes of these cross-presenting dendritic cells, leading to the failure of PD-L1 blockade. And so, ultimately, what we are working on now is to define the molecular basis of this TLR5 mediated signaling deficit in these myeloid cells. In TLR5 knockout mice treated with a PD-L1 blockade, we're working to understand why, if there's other elements that make them so much better able to respond. And so we have evidence that the T cells that accumulate over time within the peritoneal microenvironment might be more tissue resident and stem-like, indicating an ability to rapidly be deployed against tumors upon re-challenge. These are the cells that you want there during long-term to mediate long-term resistance against recurrence. And then in wild-type mice, ultimately, we're hoping to leverage the knowledge that we have gained to enhance survival and outcomes for ovarian cancer. And so one strategy that we've decided to use, because we find that these cross-presenting dendritic cells, at least in our TLR5 deficient animals, tend to associate with better response to immune therapy, we utilize an approach that has been, that has garnered a lot of attention because of the fact that in preclinical and clinical studies, if you administer this FLT3 ligand in vivo, and so inside the body, you get this really robust expansion of these cross-presenting dendritic cells. Preclinical studies have shown that it's actually pretty well tolerated. The issue, though, is that few clinical studies have actually indicated a clear benefit for using FLT3 ligand as an immune therapy. And so we tried this in our animals, and we used it alone or in combination with PD-L1 blockade. And so I'll walk you through this survival graph. And so you can see in all of the black bars that our wild-type animals received absolutely no benefit whatsoever from either the FLT3 ligand alone or in combination with PD-L1 blockade. And just as a reminder, we're using the more aggressive fallopian tube-derived model, the one which, as you can see by the solid red line, did not achieve robust benefit with PD-L1 alone. And you can see in the dashed line that is red that delivering FLT3 ligand alone also, although it improved survival some for TLR5 knockout mice, it wasn't that significant. However, when combined, the FLT3 ligand and PD-L1 blockade, we're seeing that these animals are achieving almost 80% survival in response to this highly aggressive ovarian tumor cell line. And so we wanted to know, is this survival benefit robust if we rechallenge them with the same tumor cell line in the absence of any ongoing therapies, what is their survival outcome? And what we found was really quite striking in that our rechallenged animals, there's a memory there that is able to, again, counteract these highly aggressive ovarian tumor cells, leading to extended and significant survival benefit. And so what does this mean for patients? Does this actually have clinical importance? And ultimately, what we believe then is that patients with the polymorphism could actually immediately benefit from using the DC expanding therapy of FLT3 ligand in combination with PD-L1 blockade. And in the 90% of the individuals with the ancestral allele, they might benefit from blockade of this pathway. And so does blockade show promise using our animal models? And so we wanted to test this. And so we used an antibody. It is very expensive. And so we were only able to administer it for an acute duration, 4 days consecutively at a very low dose. Really the goal determining, is this sufficient to start to transform the ovarian tumor microenvironment? And we looked at the long-term effects. And so we delivered it beginning day 15, and then we analyzed the tumor microenvironment at day 30. We use a non-biased methodology to analyze myeloid cells and dendritic cells specifically to determine whether this inhibition of TLR5 signaling affected the dendritic cell compartment. As you can see highlighted in this magenta cluster, we started to see an expansion of these cross-presenting dendritic cells. And so they're expressing XCR1 and CD103. When we go back and quantitate this using traditional gating methodologies, we again confirm that indeed there are significantly more of these cross-presenting dendritic cells within the tumor microenvironment, and they're exhibiting less evidence of being immune suppressive through reduced expression of PD-L1, really indicating that there is promise to inhibition of TLR5 signaling for the majority of ovarian cancer patients. One way that we're hoping to kind of leverage this new data, in addition to making it a little bit more specific, is to combine DC expanding therapies with TLR5 inhibition by making a bispecific antibody that's fused with the FLT3 ligand in addition to the TLR5 blockade. And we believe that this will get to the cells that it needs to inhibit TLR5 signaling, preventing the expansion of these red populations, the myeloid cells, really looking more like what we see in our TLR5 deficient animal. And so we're hoping that by doing that we can move the survival curve in combination with immune checkpoint blockade from this dotted black line to the dotted red line for the majority of ovarian cancer patients. And so in conclusion, what all of this data really suggests is that direct TLR5 signaling on CD11C expressing myeloid cells significantly impairs the response to PD-L1 blockade. And we believe that by blocking this signaling pathway, or in the absence, so for the 10% of individuals that have a deficiency in TLR5 signaling already, FLT3 ligand and PD-L1 blockade have robust potential to increase outcomes. And that's really what we're hoping will be recapitulated from our preclinical models. And then we're hoping to test the efficacy of our new bispecific antibodies in combination with PD-L1 blockade, in addition to generating a bispecific that's targeting both TLR5 and PD-L1. Because one thing, and I didn't have time to show this today, is that we find that the majority of TLR5 expressing cells within the ovarian tumor microenvironment also significantly upregulate the PD-L1, very much aligned with what we're seeing with our in vitro culture system. And so with that, I'd like to acknowledge the members of my lab. This was work that was predominantly done by a student, Mitch McGinty, who has graduated, and it's been taken on by student 3, who is really working to understand how TLR5 signaling is affecting the response at the level of the bone marrow, which is really, what we believe is kind of the originating site of this exposure to TLR5. Kara and Akshita are two new students who are also working on the TLR5 project with various different mechanisms. Akshita is looking at the metabolic aspect, and Kara is working to understand why TLR5 signaling is leading to this failure of DC expanding therapies. Audrey, Simona, Mika, and then all of our undergraduates that have been within the lab have really helped to move this project forward, either through intellectual or the large animal experiments within the lab. I'd also like to thank my sources of funding, my collaborators, and I'd like to thank you for your attention. And with that, I'm happy to take any questions.
Thank you, Melanie, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just submit a question in the Ask Question box located at the bottom of your screen. Let's get started. Looks like we have quite a few questions that came in already, Melanie. The first one asks, can this be applied to other cancer types aside from ovarian cancer?
No, this is an excellent question. And this is something that we've been working to understand. And so, and clues about this came actually through analysis of the TCGA data sets, where we were looking across other cancers for survival outcomes with individuals that have the polymorphism. And what's interesting is, for example, with breast cancer, it's actually the opposite. It's exactly the opposite. And so for individuals with breast cancer, having the polymorphism actually corresponds to a significantly worse outcome. The reasons of this could be complex. It could be as simple as just the different cells within the tumor microenvironment that are expressing TLR5. One thing that we're hoping to do, especially with the bispecific antibodies, is that this will have a broad effect, especially because we think that the pathway is being initiated at the level of the bone marrow, which is agnostic of tumor type, especially in context of solid tumors.
Awesome, thank you. The next question asks, what are the next steps regarding this research?
The next steps are to, I think what we're most interested in is kind of two-pronged. Number one, we're really curious about how TLR5 signaling is affecting these cells, especially in the context of ovarian cancer. Is it that there are unique cell types that are expressing TLR5 aside from these CD11C expressing cells? And the reason we think that is because we didn't see, the survival get to anywhere near what we saw in our full knockouts. And so we're working to understand that tumor microenvironment and the effects of TLR5 signaling really at a molecular level, in hopes that we can identify drugs or targets or druggable targets that are being engaged in this pathway to convert these cells into a more suppressive phenotype. And we're doing that through the focus on the metabolic aspects, but also functional aspects. We're also interested in understanding and developing these bispecific therapies and testing them not only preclinically, but hopefully to bring them more and closer to the clinic so that patients can actually benefit from these discoveries.
Great. The next question asks, how are bacteria entering into the tumor microenvironment?
This is a really good question, and it's something that we've worked earnestly to try to understand. And so, it could be just kind of spontaneous leakage and dissemination. It could be just, through the flow of the blood, and they're getting into the tumor microenvironment, which becomes highly vascularized, or they could be carried there by other immune cell subsets. And we've worked to try to label them with fluorescent probes, but that hasn't been, it hasn't reached the level of sensitivity to really detect on a per cell basis kind of how they're getting there. And so that's actually something that we're slowly working towards trying to address.
Thank you, Melanie. And it looks like we have a few more questions that came in. The next question is it known as to how TLR5 signaling functionally rewires myeloid cells?
No, and the single cell sequencing provides some clues, although it's not definitive, but it's suggestive that TLR5 signaling is retaining them in an immature set type, in an immature stage, but it's also leading to a stress phenotype. And the cause or consequence of that is something that we're trying to disentangle, and really kind of the next steps in the phase of this research.
Okay. The next question asks, is bacterial flagellin the only known ligand for TLR5?
Currently, it is the only known ligand, but one thing that we're doing, because no kind of host-associated ligands have been identified, we are working to generate mice that lack all bacteria, germ-free mice that are deficient also in toll-like receptor 5 signaling. And if we give them tumors and still observe that survival benefit, that suggests that there are non-bacterial ligands that might also be attributed to these survival deficits. And so that's one of the ways that we're hoping to kind of test this. But for now, we're assuming that all of the signaling is coming from bacteria.
All right, and it looks like we have time for one last question, and it is, why are TLR5 KO mice unable to benefit from immune therapy?
The TLR4 knockout mice.
Sorry,
Um,
TLR4 KO.
Yes. So that's an interesting question. And we're using the in vitro cultures to try to get at this, but also there are TLR4 reporter mice, because the simple explanation could be that the cells that are infiltrating into the tumor microenvironment are at these precursor stages within the bone marrow that are being affected, might just express more TLR5 as opposed to the TLR4. And so even though there's likely TLR4 ligands within the tumor microenvironment, they're not recognizing that. Another possibility could be that because TLR4 ligands are molecularly a little bit more distinct than TLR5, maybe their biodistribution is just completely different. And that is work that is a little bit beyond our lab's wheelhouse, but it certainly is a possibility. And so those are some of the possible explanations for that question.
Thank you, Melanie. Do you have any final comments for our audience today?
If anybody else has lingering questions, or if you think of something later, I'm happy to, just go ahead and email me. Please feel free to email me, and I'm happy to respond.
Awesome. Thank you again, Melanie Rutkowski, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 30th, 2026. Labrets will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you.